I can't stress enough when we're talking about gills and the kidneys that the epithelium is going to do a lot of the work here, right? And what you're going to see, I'm going to show you a number of mechanisms, right? And there's going to be a lot going on. But what you're going to hopefully see, and the light bulb comes on, is that a lot of the same proteins and channels and pumps are used in each of them. It's all about where you put them, okay? So with an epithelial barrier, remember, the first thing that an epithelial barrier does is it lines the surface of animals and it lines all the body cavities, and therefore is your barrier, right? It's the first barrier to whatever cavity or between cavities that we're talking about. Here we're talking about an epithelium that's exposed to the environment, right? First, we're starting with gills, which is directly exposed, right, to the water. And so that barrier, first and foremost, compartmentalizes the inside of the animal from the outside, like we talked about before. Remember, tight junctions do that, and those are apically located. The top of the cells that are sticking out towards the environment or lumen of an organ is called the apical surface. And then the part of the epithelium that's attached to the basement membrane is the basolateral surface, right? You find the nucleus, and anything below the nucleus is called the basal compartment. And anything above the nucleus, closer to the top of the cell, is called the apical compartment. So we also can use these two terms, mucosal versus serosal. So serosa is the organ side or the animal side, the inside of the organism. So when you say serosal side, we're saying basal side. That's the side pointing towards the inside of the organism. The mucosal side is the apical side because that's where we put the mucus, right? That's where we add mucus because most of these membranes are mucosous membranes. They have mucus on them. The epithelium we're going to be focusing on are those in gills, skin, kidneys, because we're going to be talking about salt transport and water absorption and loss as we do this. The basic machinery used is going to be the same, but we can customize or polarize, if you will, where we put these proteins. And as we start moving them around, it will become apparent. It's all directional, right? Which way we want to pump things. Do we want to pump things out, or do we want to pull things in, right? And all you have to do is rearrange these things. And it'll become really apparent when we start talking about fish that can move from fresh to salt water. They simply are going to switch up their epithelium as they move from one environment to the next. So what's going to drive all this is the electrochemical gradient that we've talked about 1,000 times, right? If you do it correctly, all cells, as we know, have a slightly negative intracellular leaflet on their cell membranes and a slightly positive extracellular leaflet driven by leakage channels and the sodium-potassium pump, right? So before you dive into this stuff, make sure that you know that. You can write it out. It makes sense to you so that you can take the next step, right? Without that foundational information, it becomes really confusing. So if all of these mechanisms I'm going to mention deal with the electrochemical gradient, the driver here is going to be a sodium-potassium pump, right? It's going to be found in every one of our mechanisms. And that'll be no surprise. That won't be something you have to memorize. You'll know it's there. And the sodium-potassium pump is always in these mechanisms basolaterally located. It's always in the serosal side or the basal compartment or the basal membrane side of the mechanism, pumping sodium and potassium 24 hours a day. As a matter of fact, in saltwater fish, it is incredibly important. They have more sodium potassium pumps in their membranes than any other cell ever looked at. And they also have more mitochondria in their cytoplasm than about any other cell that you've ever seen. They're called mitochondrial-rich cells because when you look at them under an electron microscope, you see like thousands of mitochondria, and it's driving these pumps, as you'll see. So why do these cells produce electrochemical gradients? To run secondary active transport, right? And so we got primary active transport pumps, the sodium potassium pump, driving secondary active transports of, in this case, we're going to be talking about ions, right? So we're going to have a number of pumps that you can see in the kidneys or gills. The sodium potassium pump is one. The other one is the hydrogen potassium exchanger, which is a anti-Porter pump that uses ATP, where you pump hydrogen in one direction and potassium in the other. And then we can just simply have hydrogen pumps that pump hydrogens, either using ATP or using ATP and a hydrogen gradient, as you will see. And these pumps, particularly the sodium potassium pump, are what drive ion movement that you're using symporters and antiporters. The other major concentration gradient we are going to use are protons and bicarbonates. So, as you're going to see, I'm going to show you the classical mechanism in freshwater gills that I learned in the 1990s that I had to memorize. And then there's that one is completely not accepted anymore. Right? That mechanism has been disproven or lost support because it's just not practical. And I'll explain why when we get there. It was the discovery of an enzyme called carbonic anhydrase that is very common in gills and lungs because it's associated with the movement of carbon dioxide. And so there's a lot of this enzyme already in the gills because it's already involved in carbon dioxide movements or carbon dioxide metabolism. And as we know, one of the major jobs of the respiratory system is to get rid of carbon dioxide, right? That you're producing because of cellular respiration and bring in oxygen. The gills are no different. They do the same thing our lungs do, right? They're extracting oxygen from the water and dumping carbon dioxide. To do that, you need carbonic anhydrase, right? And so what carbonic anhydrase does is it supports or causes the production of carbonic acid, which is this guy right here, H2CO3. And it does that by combining water and carbon dioxide together. Carbonic acid immediately breaks down into two ions, a negative bicarbonates, which the body likes to keep and use, and then protons, right? The body does not like protons, so we typically get rid of those, right? We move them. We move them into a lumen of a duct, or we just exchange them into the environment. So you're going to see carbonic anhydrase can be in multiple places. It can be in the cytoplasm. It can be on the apical membrane. It can be on the basilar membrane. It can be all over the place. All right. Another ion that still gains some supports, particularly when you're looking at dealing with acids and base metabolism, is ammonia or ammonium ions. This is an ammonium ion, NH4+. Or sometimes you see NH3, which is ammonia. And in fish deals exclusively with ammonia. They don't convert it to anything else. They just get rid of it. And the reason for that is as you're going to see towards the end of this lecture, is that to move ammonia or ammonium ions, you need a lot of water. Well, they live in water, right? So there's no reason to convert it into anything else. If I have tons of water around me, I can use that water to move it. And often what these cells will do to produce ammonia, if you got to get rid of excess base, is to do some amino acid catabolism, where we just remove the amino group off of certain amino acids, either converting them to a carbon skeleton that can be used in the cellular respiration, or converting them into another amino acid. We will then pump out that excess ammonium ion out, which will help us deal with pH changes that might happen in the blood. So this is kind of an example of a mechanism that is used in the kidneys to move glucose, all right, or in the gut to move glucose. And you'll see it in the kidneys, you'll see it in the gut. It's the same mechanism that is utilized in all vertebrates. What drives glucose, in this example from the blood into the cells, is basically a sodium potassium pump in a sodium glucose transporter, right? So by pumping potassium in and sodium out and allowing potassium leakage and sodium leakage, we keep sodium low. We use this concentration gradient to pump glucose into these cells, and then the cells can use it, right? We're going to do a very similar mechanism to this, but now we're not going to move glucose, we're going to move other ions. Okay, we're using sodium because we're interested in maintaining osmolarity, right? And so we'll look at how saltwater fish and freshwater fish do this. So again, why am I giving you all this background again? I want to make sure that you remember all your foundation, that each one of these slides makes crystal clear sense before you dive into the mechanisms, okay? That way the mechanisms are much easier. If you just dive into the mechanism without reviewing, it might be a little confusing at first, right? But if you got a hold of each of those little pieces of the puzzle as we go, we can now apply them, right? And so let's talk about the differences between fresh and saltwater fish. So we know that freshwater fish have a blood osmolarity that's much more hyperosmotic to the environment they live in. And they typically, if you're looking across animal taxa, vertebrates in particular, right? We're going to stick with vertebrates here. When we're looking at vertebrates, freshwater fish have the lowest internal osmolarities. Instead of around 300 milliosmoles, a lot of them are sitting closer to 250. All right, so they just have slightly less hyperosmotic interstitial fluids just to help deal with living in fresh water, right? It makes the osmotic pool on water a little less. But still, fresh water is typically around 50 milliosmoles. So 250 is much bigger than 50, right? And so they are always going to be taking water on, right? Water is going to be rushing in to their interstitial fluid wherever it can get in. They have impermeable skin. Their skin, their epidermis is waterproof. No water moves over their skin. They don't drink water. Okay? Now they will ingest water, right, when they eat things. But their gut is also pretty impermeable to water. That is, they don't move a ton of it, or try not to when they're digesting food. They don't need to drink water because their gills are like a water sieve. Water just pours into their gills because they're exchanging gases there, right? So the epithelium is quite thin. And so they're always taking on water. And if we don't do something about that, we're going to dilute the body fluids, right? Which we don't want to do. And so as I mentioned earlier, the kidneys have absolutely humongous renal corpuscles, right? Where the glomerulus and the Bowman's capsule is. And they basically urinate all day. They're just dumping water from the blood into the nephron and just getting rid of it. The nephron is pretty impermeable to salt, right? So they don't lose salts with those. But they do lose some salt. No matter how tight the nephron is to salt loss, you're still going to lose salt, right? And so they can get rid of water easy, but salt leaves with their urine and their feces. And they're also going to have to pump salts across their gills from water that has very little salt. And it's really quite amazing that they can do that, right? That they can remove salt at such a concentration gradient that really hinders that ability. But they're really good at it. And they've evolved these mechanisms to be really good at it. So we're going to basically learn that at the gills, we are going to pump salt from the environment into the blood to help maintain our normal blood osmolarity. And this is the classical mechanism, right? So this is the mechanism I learned a long time ago. And the biggest problem with it is this mechanism we had no idea how chloride moved. So we didn't know if it moved paracellular or transcellularly. And so you see there's no chloride on here. And the mechanism that really is driving this is the sodium-potassium pump. So the sodium-potassium pump here is keeping sodium low in the cell. That gives us a slightly negative intracellular side of the apical membrane and a slightly positive pond water side, right? and that will attract sodium. And if they bring sodium in, then they have to move a positive ion out, right? Or the inside of the cell will become positive, correct? And they'll allow sodium to obviously be pumped out again and allow potassium to escape to keep my negative charge. But if I'm bringing a positive ion in, I have to pump a positive ion out to prevent osmolarity and electrochemical gradient changes. And so the ion they thought they could move here really easily was ammonium ions from amino acid metabolism. And early on in the 70s and 80s, they actually found that the gill epithelium had the ability to break down amino acids. Right? So this was a possible mechanism. Right? And it was a universally accepted mechanism for a long time. Now, this ammonium ion is actually pumped in this mechanism, in this hypothesis. It's a primary pump. You're using ATP to pump that positive charge out to maintain this very negative environment on the apical membrane, which will drive sodium in, right? Now, the biggest problem with maintaining this negative environment are where are the chloride ions and how they get in, right? So, they thought that the chloride channels would be paracellular, right? They would be found in the tight junctions. So this is how all freshwater fish and amphibians were thought in the early 90s and 2000s to be to move salt into their bodies from pond water. But when physiologists started really looking at this mechanism, they weren't really pumping enough ammonia in living experiments for this mechanism to work. And so it was later on when they found that chloride cells had carbonic anhydrase in them, right? So carbonic anhydrase is a very common enzyme found in all of the gill epithelia. And the epithelia here is basically lined with usually one or two layers of cells that are either squamosal or cuboidal in shape. And there are two main kind of cell types. There are the really common pavement cells, which are kind of like the squamous cells. And they're involved with, oxygen and carbon dioxide exchange. And then there are these weird looking cells that have very deep grooves in them that are called caniculi, right? That are called chloride cells. And these chloride cells are named that because they discovered a chloride channel in them, right? Which was good because remember, we didn't have chloride channels before. So in our other mechanism. And so this chloride channel is a antiporter, and it took them a while to figure out what it was using to drive the chloride into the cell or to suck it from the pond water. And the ion that they ended up finding that they were exchanging had to be a negative ion, right? Because we're exchanging a negative ion into the cell, we need to pump a negative ion out, right? To keep it neutral. And the ion that they figured out was bicarbonate, right? So bicarbonate is the major anion that they're using to run this exchanger. And if that is a true secondary active transport exchanger, then it's going to need a concentration gradient to use, correct? So what concentration gradient does it use? It uses bicarbonate. So we have to be able to build up a bunch of bicarbonate in these cells, which is happening anyway because they're moving carbon dioxide, right? And so this is a byproduct of carbon dioxide metabolism. And so we can use carbonic anhydrase to combine carbon dioxide with water to form carbonic acid. And then that breaks down into bicarbonate and protons. And that will build a huge gradient of bicarbonate that you can then allow to flow into the pond water and suck or pump in the chlorides. This will create, along with the sodium-potassium pumps and the potassium leakage channel, these two proteins right here are always found in the basal membrane, right? You can almost ignore them because you know they're there, right? They're working. They're always working. This keeps sodium levels low, and it also makes the apical membrane very negative and will allow me to move sodium in. Sodium will be sucked into this because it's low and it's negative, right? And that really attracts the sodium. To really make this work, I'm going to combine this sodium channel with a hydrogen pump. So I'm going to build up a proton gradient, And I'm going to use ATP to drive sodium in. I mean, it's like a double whammy, right? It's actually a triple whammy if you think about it. It's already negative. Sodium's already low. I'm going to use hydrogen to do this. And it's like 4 different, mechanisms all in one to suck sodium out of the environment. And this makes a lot more sense because the other mechanism, it's like, oh, okay, that's a pretty cool mechanism, but is that really going to suck sodium out of water that has no sodium? If sodium's really rare and you really want it, you're going to have to really have a pump mechanism that can move it. And having really low sodium, a negative environment, and then a hydrogen gradient and an ATP pump, all working to suck sodium in, is much more rigorous, right? Much more believable. The nice thing is they found all these proteins now, right? They know they're there, or most of them at least, because they have isolated them, separated them, and found that they are expressed in these chloride cells. This also gets you sodium in and chloride in, right? Sodium's pumped out. The one thing that's missing from this picture is there is a chloride leakage channel on the basal side. Right? I didn't put it in there. But you should realize, obviously, if I don't have a chloride leakage channel going into the blood, then chloride's going to build up, right? It's going to shut down this antiporter. And so there is a chloride leakage channel here, like the potassium leakage channel, that allows chloride to leak and follow sodium back into the bloodstream. So that's how chloride cells work, right? In fresh water, and that's how they suck salt out of fresh water. Now, what's really interesting is if you go to, say, the Amazon, okay? Go to what they call a tannic river in the Amazon, which I've been in the Amazon. It's really awesome. So there are a number of rivers in the Amazon that are tannic rivers where the leaves decay and they give off tannic acid, and the pH of the waters are like 4. And I've been in them. It's really weird. You get in the water, your skin it tingles because of the pH. It's kind of cool. Plus, the really nice thing about them, there's no mosquitoes in there because they can't survive in the rivers. But the water's really soft too. It's really oily, which is really weird. So your skin's tingling. The water's really kind of freaky. Here's a goosebumps when you first get in it. Those particular places are really, really low in calcium, and so instead of moving sodium, they're actually sucking calcium out of the water because they lose calcium. And calcium is so important in nerves that instead of having a sodium hydrogen exchanger, they have a calcium hydrogen exchanger. So they're actually calcium chloride instead of sodium chloride, and that's kind of cool. But here in Indiana, our water, as you probably already know, is so hard. There's so much calcium chloride that the fish here just move sodium chloride. So just an interesting, side note, if you will, about fish. Now, this is a picture of the gills. This picture down here is really cool. And if you look, well, I killed that one light real quickly. And you can see it a little better. You can actually see the gill filaments, right? Those are the purple filaments, kind of, that run this way. And then at the base of these gill filaments, what you got lit up here are all the cells that are lighting up the hydrogen-sodium exchanger. And so they found the protein, you can make an antibody for it, and you can light them up, right? So you can see all these chloride cells, and they're quite numerous in the base of the gills, right? And so they're working really hard to add salts to, to our model here. So, make sense? Question? Yeah.
All right, so the bicarbonate moves out into the water because of the high concentration of bicarbonate inside?
Yeah, because the enzyme's always producing lots of it. Okay. So it's kind of weird. You're actually building a concentration rate without using a pump. You're using an enzyme to do it. Exactly. So this enzyme's just working all day long. It's got plenty of carbon dioxide, right? Because carbon dioxide is being exchanged between the blood and the pond water anyway. that I can grab some of it and convert it into bicarbonated protons. Does that make sense?
Hydrogen.
Same way, you get a whole bunch of it by breaking carbonic acid. This is also a very good way to get rid of acid, isn't it? The acid that's in the blood. These things produce so much bicarbonate that I've actually seen mechanisms where they put a bicarbonate leakage channel on the basal side too. Because these cells are really good at producing it. And so there's so much of it that they can run the antiporter and collect some of it. Because bicarbonates are a really good buffer in the blood supply. But if that confuses you, you can just ignore what I said. Right? Okay, marine fish have the opposite problem, right? They're going to lose water from their gills. Again, their skin's impermeable. They're going to try not to lose water in their urine. And they're going to try to push as much salt into their urine as they can, and as much salt into their feces as they can, because salt water has so much salt in it, right? And so they're going to be gaining salt all the time. And so what they want to do is, or because they're losing water over their gills, right? Because they are hypoosmotic to the environment, they're going to need to drink salt water and then convert it to fresh water. So they drink water all day long and produce very concentrated, small amounts of it, which is just the opposite, right, of what a freshwater fish does. Now, interestingly, their kidneys are very different looking in a lot of saltwater fish. So remember, when I told you about the nephron in a freshwater amphibian, right? They have huge renal corpuscles and then a renal tubule, right? This is where the glomerulus is. So their renal corpuscle, or their nephron, the first part of their nephron is really big. In freshwater fish, the nephron, the renal corpuscle will be really small or not even there. They just have the tubule because they don't want to filter water, right? They don't, because if you're going to filter the blood, you're going to filter water, right? The plasma is what leaves the glomerulus and enters the nephron. We want no water in the renal tubule because we don't want to lose any of it. So I either make a really small glomerulus and Bowman's capsule, or I just get rid of it. And then the tubule is really important in just pumping salt into the tubule and then getting rid of it. Does that make sense? And so it's really interesting when you look at two nephrons between a freshwater and a saltwater fish, and just the size difference or the absence of the first parts of the nephron called the renal corpuscle. Now, marine fish typically have the highest internal osmolarity, somewhere up around 450s in some cases, where most terrestrial organisms like us maintain about 300 milliosmoles. They try to push it a little higher so that they are not so pulled on, right, water and salt-wise, by the marine environment. But remember, the marine environment is still really hyperosmotic. It's like 1,200 milliosmoles. Right, so they're going to lose water and gain salts and have to deal with that. So notice this down here. Small amounts of urine, nearly isoosmotic, but it's very rich in magnesium sulfates and sodium chloride, which they don't show here. But we want to get rid of all of these. These are very common in salt water because they really affect the nervous system. They throw off the ability to produce action potentials. Now let's look at the mechanism. So these cells are called mitochondria-rich cells, or they're also called chloride cells, right? But I like mitochondria-rich cells. This is a drawing of what one of these cells look like. And they have more mitochondria than almost any cell that's ever been looked at in a vertebrate. And the thing they do have that, I'm talking 1,000-fold more of than any other cell that's ever been looked at, are sodium-potassium pumps, right? And so the more sodium-potassium pumps they have, the more effective they are at getting rid of salt. Okay. Now, their epithelium is set up just slightly different in the sense that these cells, these mitochondria-rich or chloride cells, are kind of pushed deep into the pavement cells and pushed into or against the blood vessel. So you can see here in this kind of mechanistic model here, we have a pavement cell on top and a pavement cell on the bottom, and we have a tight junction here and a tight junction here. And notice you kind of reset, or in a recess, push backwards your mitochondrial-rich cell or chloride cell into a little cavity, so that's kind of in direct contact with the blood supply, right? And what this allows are all of these sodium-potassium pumps to pump tons of sodium, because the sodium is just pouring across, right? Tons of sodium into the bloodstream, keeping it really low here, and bringing potassium in, and the potassium leaks out. And so what we're trying to do is keep this little cavity, the cytoplasm of the mitochondrial-rich cell, low in sodium, so that we can drive this symporter right here. Right, this symporter is the sodium potassium chloride symporter, and it has a name. This protein has a name. It's called an NKCC protein. So it's called, really what it's called is an NKCC cotransporter, which stands for sodium potassium chloride cotransporter. Okay? And there are numbers. There's like NKCC1, which is found in certain cells, and NKCC2, which is found in other cells. If I remember correctly, NKCC2 is found in gills, and then NKCC1 is found in kidneys. So these transporters are driven by a sodium gradient. And because these cells have so many mitochondria producing so much ATP and have 1,000-fold sodium potassium pumps, they have some of the lowest intracellular calcium concentrations of any cell known to physiology. And this symporter, therefore, is really effective. You can move lots of sodium in, which is going to move chlorides in. We don't really care about this sodium because it's immediately pumped out, isn't it? Right? What this creates is kind of a positive blood side of the gills and a negative ambient water side of the epithelium, because this chloride builds up so quick that it is dumped into the salt water 24 hours a day, which kind of makes the environment really close to the cell negative. And that's really important because that is going to drive sodium out of the blood through a sodium transporter here. The sodium is attracted to or follows the chloride and the negative environment of the epithelium. If you remove even half of these sodium potassium pumps in the mitochondria, then the cell doesn't work very well, right? And so what really drives this is its morphology. You know, having thousands of mitochondria and hundreds of thousands of sodium potassium pumps is what drives this mechanism, this cotransporter. It's a symporter, right? Which allows chloride to build up quickly and escape into the environment, creating a negative charge on the outside of the epithelium, which will suck sodium from the blood through paracellular transporters. And so you're just losing sodium chloride, right? That you're drinking with the water and converting salt water to fresh water. This is how every salt gland works also. So instead of having the pond here, this would be the lumen of the salt gland. So turtles, crocodiles, birds can drink salt water. Marine iguanas. Have you ever seen a marine iguana? Pictures of marine iguanas, typically they have a white head or a white face. That's not color, that's salt. Because they sneeze all day long. Their salt glands drain into their nose, and they just sneeze salt. And that's why they're always covered in salt. A lot of iguanas do this. Birds also drain salt from their nose. Some birds drain salt, and turtles from their eyes. Right? They shed tears. So Crocodilians, their salt glands are in their tongue. And so they swallow the salt and then it leaves through the digestive tract. So that's how these mitochondrial rich cells work. So this is a picture of a chloride, a transmission to a couple of different types of chloride cells. Here, or a mitochondrial rich cell. So here's a mitochondrial rich cell from the gills of a saltwater fish, showing you all the mitochondria, the pavement cells, the blood vessels down here. And that's the mechanism for saltwater fish. Okay? Or for salt glands. So here's a picture of salt glands. Works the same way. Same exact way. One difference with the salt gland is I can add an extra kind of mechanism to help move the salt really effectively. And because I'm not worried about gills and how gills are arranged here because I breathe air, because remember, you're kind of constrained with the gills because, you know, if you think about gills, the main function of gills is respiration, right? And so the primary structure of the gill is set up for respiration. And you just insert the mitochondria-rich chloride cells. But in a salt gland, I can reorganize the gland so that the fluid that's produced by the glandular ducts is running the opposite direction of blood, right? And so you've got two fluids moving in opposite directions close to each other, and we called that countercurrent exchange, didn't we? Which makes it more effective to move the salt from the blood into the duct because you can take advantage of concentration gradients. But the mechanism's the same. And this is a picture out of another physiology text that shows you the countercurrent exchange, right? Where you got the salt in the duct moving in the opposite direction of the blood by organizing the blood vessels around the elongated glands. And typically these glandular lobules, right, drain into a central duct or canal. And that duct or canal typically drains into the nose. In birds, often the gland is found above the eyes and sometimes drains into the eyes, but often the central duct drains into the nose where they sneeze out the salt. Same for reptiles and like turtles and iguanas, for example. Now, when you look at this, there's an epithelium between the blood and the lumen of the duct here, right? And that epithelium is going to be organized just like what we talked about with the saltwater fish. So here is, I blew it up, here it is. So instead of saltwater, this is the gland duct right here, right? This is the blood on this side. So notice we have lots of sodium potassium pumps, our NKCC transporter, our chloride leakage channel, and then our sodium paracycler channel, right? It's the same exact setup. And now all of the salt in the blood is moving into the gland, and then that will drain into the duct, and all that salt will leave as salty snot, right? That's the cool thing. That's why I love physiology, because I mean, this mechanism, which is found in fish, is very similar, analogous to it, right? What you see in the salt glands and marine birds and reptiles. Now, when you move to mammals, we don't have this stuff. So we have good kidneys. Our kidneys start doing a lot of this. Okay? These mechanisms. We don't have gills or salt glands, so our kidneys have to take over osmolarity functions. All right, so again, before you look at this slide, I want you to be, and listen, I'm going to ask you these mechanisms on the exam, okay? And so you need to know them. Remember, on the exam, if you struggle a little bit on the exam, if you want to get a better grade, you're proving to me that you know these mechanisms, right? And so how do you do that? Well, listen, when I didn't do well in my first physiology exam a long time ago. And I studied, but I figured out I didn't study enough. And so my professor was like, well, how do you know when you studied enough? Great question, right? I don't know how I'm going to know. He's like, when you can take out a piece of paper and draw it out. get it right. Get it all right. Get all the pieces right. I really recommend drawing a picture because pictures help you then explain it, right? And I know what you're going to do, memorize that picture, and then you're going to throw it up on the exam because you're going to all be nervous, right? That's fine, and then you explain it, correct? Right? So how do you do that? Well, get a whiteboard and draw it out, all right? Or come in here and draw, use chalk and draw on wherever you need to look, or get paper and draw. That's how we do it. The mechanisms out so that you get it right, and then you can put them next to each other and compare the two. Once you're really comfortable with how salt and freshwater gill cells work, chloride and mitochondrial rich cells, then you go to this one, okay? What's really cool is all you have to do is flip around where you insert your proteins. And really what it comes down to is some fish just completely get rid of the cells and just grow the new cell, right? Or some fish just rearrange channels within their chloride cells and pumps. And here are a couple examples. There are fish, like think of salmon, I'm using the salmon here, that obviously spend all their adult life out in the ocean, and then when they breed, they come into freshwater rivers and streams to lay their eggs. And to do that is a phenomenal feat because you typically can't throw a saltwater fish in fresh water. It will die, right? If you go out to a marine pet store and spend $50 on a freshwater tang and throw it into a saltwater tang and throw it into fresh water, it's going to die. Right? Salmon can do this. Eels can do this. There are a number of fish that can move back and forth. Now, sharks do it differently, right? But sharks can do it also. Now, what you'll notice when salmon are going to move into the freshwater streams, what they do is they gather together into large schools, and the whole school moves towards the river. And then they stop and they adjust, right? So that they move into the mouth of the river where the salinity is slowly getting less and less. And then they kind of spend days and sometimes weeks waiting to move in as their gills adjust. And then they move further in. And then finally they can, take off into the fresh water. And so what controls this switch over, right? Hormones. That's not surprising, right? They're hormones. So if I want to move from salt water, if I'm an adult, to fresh, the hormone that controls that is prolactin, right? Isn't that weird? So have you heard of the hormone prolactin? So prolactin is the hormone in mammals that controls milk production. It's named prolactin because it was found in mammals first. Guess what? It's found in fish too, but it doesn't make milk. It is used in fish to control osmolarity features in the gills. So as this salmon comes, it's in salt water and it moves up towards and closer to the river mouth, right? And the salinity is going to start to drop. What's going to happen is the amount of salt that the fish is taking on is going to drop, right? And so the first thing that happens with the drop of sodium, the extracellular sodium is going to become lower. They release prolactin. And what prolactin does, first and foremost, is shut down the sodium paracellular channel so that sodium doesn't leave. If we're going to move into fresh water, we don't want to get rid of salt. We need to now move it in, right? We need to switch up our direction that sodium chloride's moving. So we close that down and we decrease the number of chloride cells and switch over our what we call our mitochondrial rich cells, and then switch over our sodium-potassium pump because we got to slow its activity. Remember, in salt water, this is what drives. It's this pump, right? That drives my exchange, right? We don't want to pump salt out now. We want to pump salt in. Now, we're not getting rid of the sodium-potassium pumps. We're just down-regulating them, right? All right, we're down-regulating. We don't need so many of them. We're going to up-regulate the hydrogen pump, right? Because it's the hydrogen pump that drives the freshwater mechanism. And that will bring our sodium levels back to normal because we're going to start pumping sodium chloride, as you can see down here, right, into the body. And now the fish can take off, right? It can take off up the freshwater stream and it'll start gaining lots of water and urinating a lot and pumping salt. Right? And they survive and lay their eggs, and then salmon die. I mean, they die after they lay eggs and fertilize the eggs. Now, if I'm a little salmon and I hatch, I'm going to have this mechanism, right? Working full speed. I grow. I get to about 12 inches or less, and then I'm going to swim out to the ocean, right? So I can spend my adult life out in the ocean. And they're going to do the same thing. They're going to all gather together and start migrating, and they migrate to the mouth, and then they slow down, right? And start moving out towards the ocean. And as the salinity raises, 2 new hormones are going to be released into their bloodstream. Cortisol, the stress hormone, and growth hormone, which they're already producing, increases even more. Those 2 hormones are synergists. They work together to help cause the proliferation of mitochondria-rich cells and to increase sodium-potassium activity in those cells, and then reopen your sodium paracellular channel so that you can, now that salt you're bringing in, you can now get rid of, right? You can start drinking water, salt water, in order to produce fresh water. So hey, this is a cool picture here that they've done lots of studies on fish moving from these. I just threw up one graph here. So notice when the salt levels start increasing in the blood, cortisol spikes, and right after the cortisol spikes, the ability to pump sodium chloride goes way up. And so, as you can see over here, it goes way up. And this is sodium levels, right? So as the sodium levels, they continue to go up as the cortisol levels go up, and then they eventually drop back to normal after your sodium potassium pump increases. Isn't that cool? Because remember, it's the sodium potassium pumps that drive that sync order, right? That in cases. Oh, geez,